Several novel methods other than PCR have been proposed for amplifying DNA. These methods all use DNA polymerase to amplify DNA, but they avoid the high temperature requirement for DNA denaturation and the consequent temperature cycling. Which of these methods will prove useful in the long run is still undecided. Perhaps the most promising is rolling circle amplification technology, RCAT, marketed by Molecular Staging, New Haven, CT. This technique is based on the rolling circle mechanism for DNA replication used by many plasmids and viruses. Therefore, a circular DNA template is needed. This process occurs at a single temperature and does not require thermostable DNA polymerase. In linear RCAT, a DNA primer binds to the circular DNA, which is then copied many times to give a long single-stranded product that may consist of up to 1 lakh tandem repeats of the target sequence. Since the RCAT product remains attached to the original circular template, the method may be used in combination with DNA microarrays. In exponential RCAT, ERCAT, a second primer is used that binds to the opposite strand. In other words, it binds to the newly made linear strand. When this primer binds to neighboring tandem copies of the target sequence, elongation results in strand displacement. This creates single-strand branches that in turn can bind primer number 1 and so be converted to double-stranded DNA. Alternate extension using 2 primers results in multiple branching of the amplified DNA. The exponential version of RCAT can manufacture 1012 copies of each original circle in an hour and can detect a single target molecule. It is thus superior to PCR in both aspects. The major limitation of RCAT is the need for a circular template. However, variants that involve preliminary circularization of a non-circular target molecule have been developed. As with PCR itself, the RCAT technique can also be combined with fluorescence detection.